Hi, everyone. Welcome to our course. I'm Dr. Ana V. Villanueva, a physician researcher from the National Reference Laboratory for HIV, AIDS, hepatitis, and other sexually transmitted infections. My background is on molecular medicine, and today I will be discussing with you the basics of one of the newer technologies that is quickly gaining popularity among clinical laboratories, the polymerase chain reaction, or PCR. Okay, before we begin, I want to present to you the objectives for this lecture. First is to introduce the important concepts of molecular biology related to PCR. Second is to discuss the fundamental principles of PCR. And third and lastly is to present other PCR formats that are relevant in clinical diagnostic settings. So before we even dive into the more serious concepts of this lecture, let me first present to you the beginnings of this technology. PCR has been around for almost 4 decades now. It was actually invented in 1983 by no other than Dr. Carrie Mullis, a biochemist and the then lab director of the Cetus Corporation in California, USA. Cetus Corporation was one of the first biotechnology companies in the US at the time. And Dr. Carrie Mullis' team was interested in doing research on genetic causes of human diseases such as sickle cell anemia. And it was only in 1995, so you can say more than 10 years after he invented PCR, that he was awarded a Nobel Peace Prize for Chemistry. And just the trivia for all of us here who are, I know, interested in HIV research and diagnostics, as early as 1985, PCR has already been used for HIV testing in the blood. So now that we've briefly touched on the history, we can now ask, what exactly is PCR? So PCR is a laboratory technique used to amplify specific segments of your DNA. And it has a wide variety of research and clinical applications, such as gene cloning, screening for genetic abnormalities, such as for cancer and prenatal screening, and also for the diagnosis of infectious diseases. PCR is a type of nucleic acid test such that it detects genetic material rather than antigens or antibodies. So in the case of your immunoassays, we would expect that the target for these tests are, kunyari, for HIV, the following surface antigens present on the surface of the virus, or the antibodies that our body produce against these antigens. But for PCR, what we are interested in is this one inside your nucleocapsid, and definitely inside your membrane, cell membrane, your viral RNA genome. So this is the interest in your nucleic acid test. Before we go any further, I would first like to present to you some important concepts in molecular biology that are relevant to PCR. So first is the central dogma of molecular biology. Essentially, central dogma just states that all of us living organisms, we possess a sort of genetic blueprint in the form of your DNA. So this is your DNA, double-stranded. And genetic information that is present in this DNA is transcribed to RNA and is later on translated into proteins. And their proteins, we already know that they make up the structure of ourselves, of that particular biological organism. However, in some cases, some biological organisms, such as your RNA viruses, like your HIV, and the culprit of this pandemic, your SARS-CoV-2, they only exist with RNA as their genetic material. And information in this RNA is actually expressed by a process called reverse transcription. So ito siya. What happens in reverse transcription is that your RNA is reverse transcribed into your DNA. So from RNA, nagiging double-stranded DNA. Another important concept is the structure of your DNA or your deoxyribonucleic acid. So your DNA exists as a double helix formed by two complementary strands in an anti-parallel orientation. So double helix, para siyang hagdanan, helix, two strands, and then the orientation is anti-parallel to each other. DNA is always seen as, it's always read from 5' to 3' direction. 5' is where you always start. So as you can see, this strand, 5' to 3', and then opposite direction naman, yung complementary strand niya from 5' to 3'. And as you can see, as you can appreciate here, your DNA has a sugar phosphate backbone. So phosphate, sugar, it's alternating. And attached to each sugar molecule right here is either of one of the 4 nucleotide bases. So you have your adenine and your guanine. Ito siya, adenine and your guanine. They are called your purines. And your cytosine, cytosine, and your thymine, they are your pyrimidines. And always adenine pairs with thymine. They are always complementary to each other and vice versa. So your A pairs with your T, and your guanine, ito siya, guanine pairs with your cytosine and vice versa. So your C always pairs with your G and vice versa. So A pairs with T, C pairs with G, and vice versa. So yun yung tatandaan natin sa slide nito. So now that we've discussed those important concepts, we can now move on to the fundamentals of PCR. So what are the main components of your PCR reaction? So actually present in your cocktail of PCR mix, you have the following. Your template, usually template DNA, your primers, your nucleotide bases, your DNTPs, and your DNA polymerase. Last but not the least is your PCR buffer, where all of these molecules, these reagents, these enzymes, they are submerged in the solution consisting of your PCR buffer. So let's go through them one by one. So first is your template. Your template is usually genetic material in the form of double-stranded DNA, which contains the specific region that you wish to amplify. And it's usually extracted from your clinical specimen. So clinical specimen could be your blood, saliva, CSF, and the likes. So whatever specimen it is that contains your pathogen of interest, that is what you use for your PCR. And for you to be able to get your template, your DNA template, it's important to first do extraction of your genetic material. So in extraction, what happens is that you extract and purify the genetic material from your raw sample. And in the process, you get rid of your PCR inhibitors such as your alcohol, your detergents, your proteins, and others. And the most commonly used protocols for extraction of genetic material are your spin column and your bead-based extraction methods. Demonstrated sila dito sa ating illustration below. And essentially what happens is that your spin column utilizes buffers. So different buffers with different pH to allow the nucleic acid to bind to the silica membrane. So pagka iniiba natin yung pH ng ating, through our buffers, nagkakaroon ng different interactions in nucleic acid with the silica membrane. And this allows your nucleic acid to bind to the silica membrane, which can be found in your spin column. Whereas in your bead-based method, you actually exploit the affinity interactions between your nucleic acids and your molecules that are present, that are attached to the surface of the magnetic beads. And that is how we extract and purify the nucleic acids from your sample. But note that for both methods, for spin column and magnetic bead extraction, our first step is always the addition of a lysis buffer. So the lysis buffer inactivates your pathogen, and it also ruptures the cell membrane to release its contents. So if you can remember from the previous slide that I showed you of showing HIV, the structure of the virus, genetic material is inside the membrane, the cell membrane. So it's very important for the genetic material to be released from the nucleocapsid and all of its surrounding structures para ma-target siya ng ating PCR. So now we move on to primers. Primers are synthesized short single strands of DNA around 20 to 30 base pairs long. So synthesized meaning we usually order it from biotech companies, and they are specifically designed such that it would bind to your target region of the DNA. So of course, to be able to design your primers, you need to know your target region of the DNA. You need to know the sequence of your target DNA. And it's actually the starting point of your synthesis of the complementary strand of the template during your PCR reaction. And always, your primer comes in pairs, forward and reverse, because you need for each primer, so for the forward, to attach to each strand of the template. So ito yung forward, ito yung reverse primer mo. And this is the target section of the DNA that you want to amplify, this whole section. So as you can see, may complementary binding si primer mo with your target DNA. So that's why it's very important, again, to know the sequence of this region, of your target region. That is how you design your primers. And also very important is that your primers should be specific for this target region of your DNA alone. Hindi pwedeng yung primer mo pwedeng mag-bind dun sa ibang regions ng DNA, ng genome ng pathogen of interest mo. It has to be specific na dito lang sa section na to mababind ang primers mo. Okay. Now we move on to your DNA polymerase. So what is DNA polymerase? Essentially, it's an enzyme that carries out DNA replication and synthesis. So what it does is that it adds bases or nucleic acids, so your A, C, T's, and G's, to the 3' end of your primer for it to be able to elongate the complementary strand. So ito yung 5' end ng primer mo, ng reverse primer, and this is the 3' end of your reverse primer. So it adds to the 3' end to be able to elongate a strand. Itong strand na to, that is complementary to this template strand right here. Dito naman sa kabila, ganun din. This is the 5' end and the 3' end of your forward primer, and it adds DNTPs or nucleic acid bases. So yan yung A, C, T, or G. That is always complementary. Kung kunyari A itong nandito sa template segment mo, ito dapat T. And then dito kunyari G, dapat C ang i-add dito ni Taq polymerase. So it's very important na yung Taq polymerase mo, since it's an enzyme, it's quite sensitive to degradation. So very important na we maintain its integrity during our PCR preparation. And the most commonly used enzyme is your Taq polymerase. So Taq polymerase is a kind of DNA polymerase that is normally found in bacterium isolated in hot springs. So why this particular enzyme was selected is because of it's resistant to heating and cooling cycles that we will know later on is the usual setup in your PCR cycle. Maraming heating and cooling phases throughout the PCR cycle. And then lastly, nabanggit ko na rin kanina, you have your nucleotide bases, such as your, it's your deoxyribonucleotide triphosphates, DNTPs for short. So you have your adenine, your guanine, your cytosine, and your thymine. So these are synthetic oligonucleotides that are free-flowing in your PCR mix for your polymerase to be able to access them and to be able to add them to the growing complementary strands. So sila yung dinadagdag ng Taq polymerase para magkaroon ka ng complementary strand. And the result, the end product after this elongation process is this one. Ayan, meron ka ng complementary new DNA strands. So complementary siya ng iyong template strand. Itong dalawang template strand na to. Okay? And last key player, last but not the least, is your PCR buffer or your reaction buffer. So what is the PCR buffer for? PCR buffer essentially maintains the stability of your PCR mix throughout the stages of the PCR cycle because it gives that right pH balance in your PCR environment regardless kung nasaang stage na siya ng PCR cycle mo. And it also keeps the PCR, the pH stable enough. It maintains the pH stable para hindi ma-degrade yung mga molecules and yung mga enzymes that are involved in your PCR cycle. And also, one of the very important components other than your buffers, one of the very important components of your PCR buffer is actually your magnesium chloride. Why? Because magnesium is actually a cofactor for Taq polymerase. So without magnesium in your PCR buffer, hindi gagana si Taq polymerase, which is why in optimization of our PCR, bago tayo mag-conduct ng mga experiments or runs in the laboratory, this is actually the levels of your magnesium chloride. It's actually the one that is commonly being changed for us to be able to optimize our PCR runs. Okay. And then here in this slide, I'm just trying to show you the various stages of your PCR cycle. So we actually have three stages in our PCR cycle. First is the denaturation, second is the annealing, and third is the synthesis, otherwise known as the extension. And they all work in differing temperatures. So first is your denaturation. What happens in your denaturation is that your double-stranded DNA are heated. Usually it's very high heat, as high as 95 degrees Celsius, to allow for your double-stranded DNA to actually separate from each other. So itong heating na to, it destroys the bonds between your double-stranded DNA para maghiwalay sila. And then this step is followed by your annealing. And annealing, usually it's between 55 to 60 to 65 degrees Celsius, depending on the design of your primers. So like what I told you a while ago, primers are synthesized and are specifically designed to target the region of the DNA that you're interested to amplify and to to detect. So in your annealing, what happens is that given this temperature, it allows your primers, your forward and your reverse primers, to actually anneal or to bind to the flanking regions of the target DNA. So nagbabind sila doon sa complementary target region ng DNA mo. And the next step is your synthesis or your extension stage, which occurs in 72 degrees Celsius. Ito medyo uniform yung temperature nito, 72 degrees Celsius. So what happens here is that your DNA polymerase now attaches to the primer and extends the 3' end of each primer along the template strand. Ayan. So, ina-extend ni Taq polymerase by adding your DNTPs. Like what I said earlier, the addition of your DNTPs is always complementary to the base that is present in your template strand. So, kapag A dito, ang ma-add is T. And then if C dito, ang ma-add is G, and so on. So, for both strands, nag-elongate yan at the same time. And this is the resulting product. So you have now your two original, your template strands, and your two new DNA strands produced in one PCR cycle. So these steps are repeated in cycles of 25 to 35, sometimes even 40 cycles to exponentially produce the exact copies of your target DNA. So even if you have, say, just beginning with 2 copies of your template DNA at the start of your PCR, you can have, because of exponential amplification that occurs in your PCR, you can end up with up to 10 to the 9th copies at the end of your 30th cycle because of ito ngang exponential amplification na tinatawag. So during your first cycle, you only have 2 copies. At the end of your second cycle, you have 4. At the end of your third cycle, you have 8. And at the end of your 30th cycle, you have 10 to the 9th copies. So you can imagine even if you have very low starting material at the beginning, you can multiply the number of copies through changing the number of cycles in your PCR. Okay, now we proceed to the different formats of PCR. So yung kaninang pinresent ko sa inyo, that's your conventional polymerase chain reaction. But sometimes it's not enough. Sometimes we need other formats. So I'll present to you now reverse transcription PCR, also called RT-PCR, which uses RNA as your starting material. So RNA as your starting material, parang familiar, saan siya commonly used? So sa mga, if you want to detect RNA viruses, for example, like your SARS-CoV-2, your HIV, dengue, you have RNA as your starting material. So what's different between your reverse transcription PCR and your conventional PCR is that you have this you have these two additional steps at the start of your PCR process. So you actually have also this key player called, it's another enzyme similar to your Taq polymerase. It's called reverse transcriptase. So reverse transcriptase is a RNA-dependent DNA polymerase that can produce cDNA or complementary DNA from your template RNA. So let's look here sa diagram natin. So we have the starting material, which is your RNA, and then we produce the complementary DNA strand with the help of our reverse transcriptase enzyme. So para lang siyang si Taq polymerase, it also utilizes, it needs a primer. Again, primer is specific to your target region of your RNA naman. And then it's actually the reverse transcriptase that adds DNTPs. Again, introduced na kanina si DNTPs, si A, C, Ts, and Gs that are added, again, complementary to the template strand of your RNA naman this time. So at the end of this cycle, you already have your cDNA RNA hybrid formation. And if it looks familiar to you, it's because it's now double-stranded, just like the starting material in your conventional PCR, which is a double-stranded DNA. So now that we have this double-stranded template, we can proceed with the remaining steps of your PCR na similar lang sa ating conventional PCR. So there you go. And now we will also discuss real-time polymerase chain reaction. Ano naman tong real-time PCR? It's also known as your quantitative real-time PCR. And it involves the use of probes. So essentially, what is a probe? Probe consists of your synthesized oligonucleotides, your fluorophore, or also known as your reporter molecule, and a quencher. So again, pag probe, you have your oligonucleotides, fluorophore, and a quencher. And the detection and measurement of fluorescence from your reporter or your fluorophore after the hydrolysis of your probe during the PCR process is what actually gives out your result in real-time PCR. So let's go through this diagram. So if you can see, very familiar process lang, heat denaturation muna and then primer annealing. But this time you have probe hybridization that occurs together with your primer annealing. And then last is your extension reaction. So denaturation, annealing, and extension. But what is the difference in your real-time PCR compared to your conventional PCR? So for first step, heat denaturation, still the same. In denaturation, maghihiwalay pa rin your double-stranded DNA template. And dito papasok yung difference sa primer annealing because together with the annealing of your primers at this step, you also have annealing of your probe or the hybridization of your probe. So as you can see, medyo katulad ni primer si probe. In a sense, na si primer, di ba, it has synthesized oligonucleotides that are designed specific to target your DNA of interest, your DNA region of interest. Ganun din kay probe. In a sense, na you also design it to be able to anneal, to be able to attach or bind to your to a region of your template. So for example, ito yung region niya. But the thing is, your probe comes with a fluorophore or your reporter molecule and a quencher. So habang magkadikit sila, there is no fluorescence emitted at this time. Habang magkadikit pa sila. Ayan. But it's during your extension reaction. So this is your very familiar Taq polymerase. During the extension reaction, once the Taq polymerase elongates the complementary strand, it also hydrolyzes your probe. So na-hydrolyze, napuputol yung interaction ni fluorophore with your quencher molecule. And what happens is that kapag nag-hydrolyze na ang probe mo, what happens is that the fluorophore actually emits a fluorescence that is measured during your real-time PCR. And this fluorescence, the intensity of it is measured. Kaya ang tawag sa kanya is reporter molecule. And this intensity is what is recorded later on, makikita natin dun sa ating amplification curve during your real-time PCR. So what are the advantages of real-time PCR? For one, it's quantitative because it can measure amounts of your starting material. And two, it's real-time. It reveals partial results during the PCR run. And three, it is more sensitive than your conventional PCR. So to your right is actually the graph that is presented to you in your real-time PCR. So this graph essentially is ito on your x-axis It's the number of cycles during your run, and on your y-axis plotted is the relative fluorescence units, or the RFU, which is the fluorescence intensity scale. So gaano ka daming fluorescence yung na-emit ng fluorophore or ng reporter molecule mo, it will be reflected on your y-axis. And plotted are the amplification curves. So ito yung amplification curves of the samples and controls. So as you can see, ito si sample 1. How you interpret it, maybe a lot of us are already familiar with it. The lower the CT value or the cycle threshold value, no, the lower the CT value, that means na mas marami yung starting material mo of your pathogen. And the higher the CT value, that means the higher the CT value, that means mas konti ang starting material mo of your pathogen. So, like what I mentioned a while ago, real-time PCR is quantitative, such that you can measure amounts of your starting material given your CT value. And it's real-time because as you go through your PCR run, itong mga amplification curves na to, they begin to show on your screen, on the screen of your PCR machine. Okay. And of course, it's faster than your conventional PCR because you already have your results easily accessible to you, whereas in conventional PCR, usually how you visualize or how you interpret the results is you make an agarose gel and you perform agarose gel electrophoresis, and then you visualize this gel on a UV transilluminator for you to be able to see the results of your conventional PCR. So in general, the advantages of your PCR over your traditional methods are the following. 1, you have better sensitivity. Number 2, you can detect all organisms as long as you know the DNA sequence of these organisms, and as long as you know where you can actually purify them, from what clinical sample you can purify them. And number 3, you know that PCR is also fast and easy to perform compared to your conventional methods. Kaya lang, what are the disadvantages of your PCR? Because it's sensitive, it is very prone to contaminants. And PCR will not be able to detect if your pathogen that you detect is viable, or if it's still alive, or it's non-viable, if it's already dead and it's just remnant genetic material that is being detected in your run. And lastly, you also have to consider that there are a lot of inhibitors that can affect your PCR run, and these inhibitors are present in your clinical sample. So you have your proteins, your fats, and anticoagulants such as your EDTA or your heparin, which are present in your collection tubes. So ito namang inhibitors na to, they can be addressed by good laboratory practices and by selection of proper and appropriate high quality genetic material extraction kits. So before I hand you over to the next lecture, let's summarize what we have discussed in this presentation. So first, I introduced to you how PCR came about in the 80s. And I also mentioned very important molecular biology concepts such as the central dogma of molecular biology and your DNA structure. And third, we also discussed the components and processes involved in your conventional PCR. And then fourth, I presented the reverse transcription and real-time PCR formats and their applications. And lastly, I discussed the advantages and disadvantages in general of your PCR technique. So with that, I hope you learned something new today, and We're excited to have you during the question and answer. So ipunin niyo yung questions ninyo about this lecture, about this presentation, and see you on our live question and answer. Thank you for your attention.